Thank you all for joining us for the Dana Farber Harvard Cancer Center Connect Science seminar series. I'm absolutely delighted to introduce my colleague, Peter Sorger, who will be giving today's talk. Peter is the Otto Kreyer Professor of Systems Pharmacology at Harvard Medical School and the head of the Harvard program in therapeutic sciences. He received his PhD from Trinity College in Cambridge, UK, and then was a postdoc at the University of California, San Francisco with Harold Warmus. Before coming to HMS, Peter was a professor of biology and biological engineering at MIT. His research focuses on understanding the signal transduction networks that control cell proliferation and cell death, and how dysregulation of these networks can cause diseases like cancer. He's also has a focus on designing therapies that can target these signaling proteins. His group uses both mathematical and experimental approaches to construct and test models of signaling. They've also recently developed exciting new technologies for measuring cell states, especially in situ without disrupting the spatial context of the cells. I think he will talk more about that today. As the head of the Harvard program in therapeutic sciences, Peter also leads a university-wide effort to advance the basic and translational science that's focused on developing new therapies, engaging their efficacies in clinical trials. So I'm very excited to hear Peter's talks. If there are any questions, please post them in the Q&A tab and not in the chat. At the end of Peter's talk, I'll convey the questions to him. Peter, please take it over.
Sahan, thank you very much. Thank you for that generous introduction, and thank you all for inviting me and for coming today. It's a great pleasure to be able to talk to all of you, my colleagues, about some work we've done recently on looking at the spatial distribution of immune environments in human cancer. So I want to start just by mentioning the key collaborators in this work. Above all, Sandro Santagata, a pathologist in the DHFCC and at the Brigham and Women's Hospital. And you'll see a whole host of pathologists here. So this is really, today is going to be our foray that's been about 3.5, 4 years old in an attempt to bring increasing molecular precision to bear to the type of histopathological analysis with which we're all familiar. And that's going to be based on highly multiplex tissue imaging in various forms of transcript profiling. And I'll mention these people as we go along, but we're enormously grateful for their contributions. And we've also had a whole series of collaborations with oncologists and surgeons in Brigham, Dana Farber, and Mass General Hospital who've helped provide us with specimens and also with critical questions that we seek to answer. So before going, let me just say I have a series of relationships to disclose here. The ones that are relevant are in yellow, and that's my being on the board of a couple of companies whose technologies I'll show you. And in each of these cases, they're NCI-funded efforts to develop new methods that can be applied to spatial tumor profiling. So really, I have three things that I want to talk to you about today. The first is sort of the state of the art, in a sense, of spatially resolved tissue profiling, along with recent developments and current challenges. I want to talk about this whole issue of statistical power that's emerged. How do you know whether you have a large enough specimen, and what can you find out from that? And I'm going to compare the data you can get from tissue microarrays or selected field of view with whole slide imaging, which is the diagnostic standard, and then some 3D reconstruction. And then I want to give you a sense of the kind of underlying biology that we can get at from tumor immune interactions that we've been looking at in early melanoma as part of the Human Tumor Atlas Network set up under the Biden Moonshot Initiative. The HTAN has as its objective to try to build online atlases of histological and molecular data that in many ways are sort of the analogous information to what we got from TCGA, primarily from sequence. And the idea is in this part in particular to go from cellular information, single cell spatially resolved omics up to histology, embed that ultimately in radiology type images so we can actually get oriented within the anatomical specimen, and then use that both to get enumeration of cell types and states, but also to be able to get quantitative description of recurrent spatial arrangements. And if you're interested in the motivation of this and a first pass look, there's an open source paper on this that was published last year at the outset of this effort in Cell. And so the notion is, as I mentioned, this enumeration of cell types and programs and their tissue context and sense will provide the information for understanding tumor evolution and tumor networks that we get at a dissociated level from TCGA, but that it'll also provide insight into new diagnostics that might be translated into the clinic. And as you know from the work of Scott Roddick and others, it's now feasible to perform multiplex imaging, for example, to try to get immunophenotypes that might be responsive to ICI therapy. And we hope to actually add to that knowledge base and actually move those relatively quickly forward into clinical development. So the approach that we've been using to construct these sorts of atlases and to get insight from them start typically with pathology or radiology guided specimen selection. We then use multiplex immunofluorescence that I'm going to tell you about, a public domain method that we've developed. Conventional histology remains all critical, particularly when you consider that standard diagnostic criteria are often established through clinical trials with hundreds or thousands to tens of thousands of participants. So a difficult resource to recreate at the moment with omic methods. We do spatially resolved RNA sequencing. We do some tissue mass spectrometry and then try to do integrated data and analysis. And for much of what we're interested in, the primary specimens, and I'll mention this in particular for melanoma, are available only in fixed form, really as a diagnostic necessity. And so our work overwhelmingly emphasizes the type of data that can be acquired from conventionally paraffin embedded and fixed specimens of the type that you could get access to through a standard pathology workflow. And that then presents us with an additional challenge and opportunity, and that is the fact that as currently practiced, clinical pathology is still fairly pre-digital. A picture from the MGH here on the left in the 1960s, and I think from Sandro's desk on the right, in which you can see there's still physical manipulation of glass slides underneath a transmission light microscopy. And the question is, can that now be complemented or even supplanted by the use of digital methods? And I think we'd like to see the development of research practices that are congruent with this introduction of digital histopathology in a diagnostic setting. There are two sort of distinguishing ways we can think about doing the analysis that I'm going to show you today, and this derives from thinking about what's done in histology. And one of them is sort of a kind of bottom-up analytical method. And you're going to see this if you go to the existing literature as the dominant notion. And it's the way we might do single-cell genomics, for example. You'll end up with populations of cells here, a result of image visualization. You'll get microenvironments and social networks, and from that you'll derive features. And the attempt is to use those features then to understand cohorts of patients. And I'm going to argue today that at the level of power that we have, both in terms of spatial and number of patients and also the way we analyze images, this is in fact extremely difficult. And it remains difficult to try to argue from any particular spatial feature that you do or do not have a feature that's representative of any particular cohort. The alternative approach that I am going to emphasize as an alternative, at the moment, is this kind of top-down analytic approach in which we actually start with graded features that are actually called out by standard histopathological analysis. And here you can see this is from a colorectal adenocarcinoma, sort of drawing on the top, and then H&E images from below. And we then take this information and we use that to guide the analysis of the high-plex information. And in particular, we then can take the H&E as sort of an intermediate between the standard staging information that we would get, for example, from the AGCC type manual, and add to that molecular information. So the logic here is to build then a high-plex and spatially resolved tissue imaging infrastructure on top of the existing infrastructure for pathology, sort of as an intermediate towards a longer-term goal of actually running clinical trials to look at new diagnostic features that derive directly from high-plex analysis. And we refer to this approximately as a kind of top-down analysis. So let's look at what sort of the state of the art is in this kind of multiplex whole slide imaging. Here's a picture of a normal colon that we took fairly recently. We typically can get on the order of 20 to 60 plex images. 60 is what we claim, although often we've run out of good antibodies to think about, and when we get around 20 to 40. What you're seeing on the left side of this, upper left side of this picture, is a whole slide image about 2 centimeters square. So it's going to have around 10 to the 6 to 5 times 10 to the 6 cells in it. Here you see the intermediate resolution of each one. That little gridding you're seeing on the left, those are individual tiles, each of which is a full resolution megapixel image. And you can just now on a computer zoom right down to single cell resolution and begin to look at individual immune and epithelial cells in this case and get their census. And the critical features in that are not only the plex of the assay, which is what you tend to hear emphasized in these sorts of presentations, but also the sensitivity, the optical resolution. I'll show you an example of that. And also in section 2, this issue of spatial power. And of course, none of this is going to make any sense unless you have the right cohort and the aforementioned histopathology annotation. I'm often asked what of many of these methods are best. There are at least 8 extant methods for doing this. I'm not going to tell you about most of them, but there is a project, NCI-funded project right now across 5 centers, 5 active centers and 3 in an analytical mode, looking at the vast majority of the published methods and trying to come up with comparisons, and that'll yield results later this year. So this gives you a sense of now what a tissue image that we might acquire in our lab looks like, and I'll show you a little animation here. This is going to be of the melanoma specimens, the primary melanomas. I'm going to talk about a little later in the talk. You're going to see a small 3D visualization, and then you're going to see us moving around in this specimen to try to get access to regions of particular interest. So we typically will start off with this whole slide image, a couple of square centimeters, as I mentioned, around 10 to the 6 cells. We'll look for regions that appear to be of particular interest. Those can then be reconstructed in 3 dimensions using optical reconstruction microscopes. Channels are being switched on and off here just to make it easier to see individual cell types. We can then put different markers on top of each other, look at this inspectionally as we're seeing here, but also enumerate this computationally, and then look in particular for features of greatest interest. So here we see a SOX10 positive tumor cell melanocyte that's next to a T cell in which you can actually see sorting of the TIM3 and LAG3 checkpoint protein. So that's the kind of feature that we're able to ascertain from these and then relate back to underlying molecular programs. And this gives you a sense of what that would look like at the highest possible resolution achievable in a subset of the specimens. And it also gives you an example of some of the challenges of this type of imaging. So what I'm showing you on the right now is a CD163 positive macrophage, and you can see this sort of extension as it reaches over towards a tumor cell. And we think, and what you're seeing in the little picture here is a PD1-PDL1 interaction. So we actually think this is an immunosuppressive interaction. I said tumor cell, but I meant to say a T cell here. And here you can now see that once again in a sort of 3-dimensional reconstruction. This is the primary data. We've now gone and actually done volumetric rendering in here. And you can see that here, this macrophage is actually reaching on the order of 20 to 50 microns through and around multiple other cell types that are not visualized here to a T cell to make an immunosuppressive interaction. So ultimately, these sort of kind of detailed features are the ones that we want to put in place alongside the large-scale enumeration of cellular environments. We've also managed to move this technology through a bridge project, actually a footbridge project, which is its antecedent, into genetically engineered mice models with the work of postdoc Giorgio Gaglia and the JAX lab. And that panel is becoming increasingly sophisticated and allows us to actually make focused hypotheses and then begin to test them. And as a general rule, I think one of the things we see is how much simpler and less crowded these gen models are than actual human tumors. And we'll have much more to say about that over the course of the next couple of years. Now, as we do this type of imaging, we're almost always wanting to relate the features that we've seen, those morphological cell biology features or the histological ones, to underlying molecular programs. And here the challenge we have is getting transcript data from FFPE specimens. And we use two methods. One of them is commercialized by NanoString, the so-called genomics digital spatial profiler. But here's one we developed in-house that I thought might be of interest. And here we actually use microneedles. This was developed by the company Rareside in an STTR grant that we have with them, a small business grant. And these microneedles, which are the same type of needles that are used for semiconductor fabrication, can go in like a sewing machine and actually pick their way across regions of interest. And here we're once again looking at one of these melanomas, pulling out individual specimens, depositing each one, and then subjecting them to sequencing. And here you see a sort of simple PCA of the underlying, what we call micro regions of interest. So we call this micro region transcriptomics. We can get down as small as about 200 microns with the NanoString method and down to about 20 micron radius with PIC-seq, which is a couple of cells. And here you can see that you can actually pick out dramatically different features as you move around in the tumor, as you might expect, and you can do gene set enrichment on them. And this has been sort of critical for our discovery and allows us then to take many specimens. Each of the rows here is a different specimen, and then take a staging of these things and look across. I should have said each of the columns is a specimen, and then we can look at various programs here. That's each of the rows, and then we can relate them between different stages of disease that we can find within a single tumor. So this will contribute fairly substantially to the story I'll tell you later, looking at programs of immunoinvasion in melanoma. And the last method that we've developed for this is multiplex mass spectrometry. This allows you to do discovery mass spec on FFPE specimens. At the moment, it's a scraping technology, or actually you cut the scroll and directly put it into a tube. We can't yet do much resolution, although there's work in that direction. We can do 16 samples at a time, label them and mix them, and then you run them in the mass spectrometer. This we've primarily done because of the shutdown we all just endured in lung tissue that we got from COVID specimens. But the thing I want to point out is we can get on the order of 7,000 proteins quantified, which is sort of nice. And one of the nicest aspects of this is that you can look at aspects of signal transduction that are not generally visible by an imaging assay. And what I'm showing you here are the cytokines themselves visualized directly in human tissue, CXCL5 and interleukin-1 beta. We can look at the interleukin-1 regulators, and then downstream at the levels of NF-kappa B, and also at phosphorylation on these proteins. So ultimately, I think these three types of data, spatially resolved imaging, spatial transcriptomics, and what currently is sort of a bulk whole slide method from proteomics, but will get, I think, increasingly resolved over time, are going to provide highly complementary insight into the immune environment in individual regions of tumors and adjacent normal tissue. Now, the biggest challenge that we faced in this is not actually the methods I've just showed you, but is data analysis. And I appreciate that much of this is plumbing, as I'm showing here. But over the past year, the largest effort we had, and it didn't suffer at all from having been shut down from COVID, was to actually build a whole set of tools that allow you to go from a whole slide image, which could be on the order of a 3D data set I'll show you later is as large as 1 terabyte, down to individually identified cells that you can then analyze using familiar single cell technologies. And this is available at MC Micro if anybody's interested, a public domain method coming out in Nature Methods in the next couple of weeks, and consists of a whole series of algorithms that have had to be custom built for this. So we have all of this ready for us for genomics, thanks to the effort of the Broad and many others, but not yet for imaging. And the last piece to say is we still have quite a lot of work to do on quality control. Even the best preserved specimens have strange artifacts in them that we're only starting to understand. And it might only affect 5% of the specimen, but machine vision systems find the outliers preferentially. And so one of the things I would recommend anyone doing this is you have to inspect the images closely yourself. You can't simply rely on these automated software tools. And then finally, there's there's a whole data, a sort of visualization challenge. Because remember, we would like to see derived data in light of the underlying information. And for that, we've been working on a series of methods with Hans-Peter Pfister's group over at the School of Engineering and Applied Sciences. This is the work of Robert Kruger here. And what you're seeing is a high-plex image. And then you have this sort of lensing feature on top that can then show you individual cells that have been found by the computer. And we're trying to work out how to now overlay transcript and proteomic data on top of it. And then once you found something of interesting, you compile it up over on the right in one of these sorts of galleries. And so this itself turns out to be sort of a challenge that really is at the state of the art with what we can do with machine vision or AI systems. I showed you sort of an exploratory visualization. We'd like to get to the stage over the next few years where you could say, hey, that looked pretty interesting. I'd like to save that kind of spatial arrangement, and then I'd like to find examples of that in all other images which I have looked at, or for example, that might be found in one of these online atlases. Imagine that as the analog of gene set enrichment. And that's exactly what Hans-Peter Pfister and his group, and in fact, actually an expanding group at Harvard Medical School is now working on. And that's going to involve humans and machines working closely together, which, as many of you will appreciate, is an area of a lot of interest in contemporary applied mathematics and in artificial intelligence. And then finally, we have this challenge of data distribution. Many of you may wonder, can I get access to the data that I'm showing you today? And the answer is yes. If you email me, we can, and I'll show you how that works at the end of the talk. You can just zoom around and look at some of this information yourself in a browser. We're working with we and many others are working with the NIH to try to build some kind of a data commons that you can get access to this. Because we have to remember, all of the pioneering work in genomics really relied on standardized data sharing. And yet data sharing in digital data sharing in pathology and in this type of imaging does not yet exist. So we're investing in the data, but not yet in working out how to share it. And that in itself represents something of a technical challenge. So I'd like to tell you now, with sort of a bit of this background on the methodology, the insights from 2 tissue atlases, both of which can be accessed in bioRxiv. That's mandatory for projects that are supported by a Moonshot project. The second one is in revision for Cell, and the top one is in review. But you can find them on bioRxiv. And in this case, I want to start off by looking at what it is we could actually get from a study of a fairly large specimen and thinking about how much of the specimen we have to image in order to understand it. So what you're looking at on the left from this colorectal cancer, and we've looked at this point at around 100 of these specimens in tissue microarrays and about 40 of them in whole slide images, and I'll give you a synopsis of that. These are fairly complex specimens in some cases with multiple regions of tumor invasion. Those are labeled A, B, C. You can see one of the sort of features of the high-plex image, just a couple of channels over here, and that allows you to do through MC Micro, which I showed you on the sort of standard enumeration and find immune cell types, stroma, et cetera. This specimen is around 10 to the 7th cells, what I'm showing you here, around 10 to the 9th pixels. And we actually collected it in 3 dimensions. So we have 25 serial sections, and each of those is an H&E section and then a SciSif section. And it allows us to ask what the relative value of 3-dimensional reconstruction is over 2D imaging. This gives you a sense of that data set. And then on top of it, we could build both virtual, so computed and real tissue microarrays. And the tissue microarray that was put together by Sandro and his colleagues involves 93 patients, each with 4 cores. And we, in many cases, will have the matching whole slide image for those cores. And we'll have done that, in this case, for 25-plex imaging for the total data set. And then we have about another 100 antibodies or so that we've looked at on selected regions. Now, one of the first things we looked at here was the size of a typical feature. And what you're seeing on the left and the right is real space, X position and Y position. Here's real dimensions, so about 2 centimeters on a side. And this is now a computed version of the position of CD31 on the left or CD20 on the right, an epithelial cell marker, an endothelial cell marker on the left, and then a B cell marker on the right. And the blue here represents various regions of histological annotation. And one of the things you can see is that these structures are actually in many cases fairly large, and we can get a precise enumeration of that by looking at spatial correlation. And that is to start at any single point, for example, that's keratin positive, and then ask what is the likelihood that we have a keratin positive cell within some distance of that initial seed. And you can see it's different for keratin, alpha SMA, and FOXP3. And if you then look at these sorts of markers, go back again. If you then look at these sorts of markers in space, you'll see that that actually corresponds to regions that you would expect. CD20 marks a tertiary lymphoid structure. It's around 500 microns in length scale. CD31 will mark the microvasculature, and it's on the order of 80 microns in length scale. So we're simply getting out from this kind of simple statistic exactly the sort of thing that makes us interested in tissues in the first place. But the warning sign came when we looked at this, and you can see that the spatial length scales here in many cases are on the same order as the size of a typical sample. So here's a tissue microarray core, and they vary in size, but they're on the order of 1 millimeter or so. Here is a field of view from a method, these mass spec-based methods like BIVI or IMC. And as you might appreciate, sampling a specimen on the same scale as the characteristic feature is going to be problematic. And to illustrate that, we did the following experiment. We took the whole slide image that I showed you here. So this is all of this colorectal sample. We then subsampled it using virtual tumor microarray. So we just went in and had the machine find a keratin positive region that was a TMA, and then we did a pathology review and said, yeah, that's a reasonable TMA that I might construct. And I've showed you a couple of those cores. And then we went and just did something very simple. We calculated the abundance of a keratin positive tumor cell, FOXP3, large immune cells, alpha SMA, part of the stromal cells. The actual abundance is shown here by the arrow. So that's a complete enumeration of that two-dimensional section. The dots here show you what happened if we looked at all of the three-dimensional sections that we had. So that gives you some sense of variation within the specimen. And then the region here labeled as VTMA, so the large scattered dots, that's what happens if we look in the virtual tumor microarray. And so what that tells you is if you subsample the largest feature, which are here keratin positive cells or lumps of tumor, on the same scale as a tissue microarray, your abundance can vary all the way from 0 up to about 98%. So essentially, you have no determination of the actual abundance, which in this case is, I think, 42%. We were able to compare that to tumor variation by looking both at TMAs and whole slide samples. And the error in subsampling a large specimen with these little fields of view is as great as the difference observed between individual patients. And it turns out that this has been known for a long time. We rediscovered it empirically, and it's known as the central limit theorem under correlation. So we're all familiar with the notion of how many things we have to sample from a well-mixed specimen, flow cytometry or single cell sequencing. Our gut tells us we need on the order of tens of thousands, thousands to tens of thousands of cells to get down to an error of around 1%. But in the case of correlated signals, anything you're going to see in an image, you have to think about what's called the effective N. And that scales based on the length scale, L here, of the correlation. The practical matter is something as simple as accurate enumeration of cell abundance needs whole slide imaging and on the order of 10 to the 6 cells or more. So 100 to 1,000 times more specimens than you would need if you were doing a well-mixed specimen and looking at flow cytometry, for example. Having found that, we then we then realized that there was going to be value in looking at 2D images. And in fact, most of what we do now is 2D imaging. But we have now done some 3D reconstruction. I want to show you an example of that with a focus in particular at the invasive tumor margin of these colorectal cancers. And what's been known now for about a decade is there is an epithelial mesenchymal transition involving the familiar players seen here on the left that occurs, and it gives rise to this phenomenon known as tumor budding. And tumor buds are of prognostic and diagnostic significance. Here's an example in H&E on the left, hard to see, easier for me to see on the right, where they've been stained for cytokeratin. And the criterion for a tumor bud is that it's a cluster of cells separated from the main mass and has 4 or fewer. So it's just, think of it almost as a kind of little vesiculated structure that's broken off the tissue and is seen as being invasive into the underlying normal tissue, in this case, or into the muscle. So we did a 3-dimensional reconstruction of this. And lo and behold, what we found is what appears to be small individual vesicles when looked at in 2 dimensions is in fact a large interconnected cellular structure. And I'll show you a movie representation of that in the left. But here are all of the buds that were annotated by a pathologist in red. Here are sort of additional buds that the computer helped us find. Here's the best we can do at 3-dimensional reconstruction here. So not entirely satisfactory, but what it shows is buds which are annotated here in green and purple are in fact connected directly to the tumor mass. So what's happening in a bud is not actually a set of vesiculation events, but instead think of these as fingers that extend. There are a whole series of fingers that extend out from the normal margin. They're completely contiguous with the whole tumor. They're shown here in these colored reconstructions. And what 4-cell diagnostic criterion is simply one of these fingers in cross-section. And we're now able to then measure the EMT transition along a finger and show that this is a continuous transition from a relatively epithelial type phenotype out in the tumor mass to the mesenchymal phenotype at the very, very end of the finger. And it remains uncertain from our work and now some recent work of others whether in fact there are any isolated vesicles at all, or whether in fact these are 2 large 3-dimensional structures invading each other. We've looked at this phenomenon also from with respect to the mucin pools here, and we find that those are also large structures, and they're also interconnected and contiguous with the lumen of the gut. So characteristic of these 3-dimensional reconstructions is the discovery that many of the things we think of as small in section are in fact large. They just are connected in the 3rd dimension. And we're learning to work out how that is, how that actually can be done more efficiently than what I showed you today, which was the result of a couple of months of work. The second thing we realized in this analysis, and it pertains also to the melanoma, the last part of the talk I'll show you in a second, is many of these phenotypes are in fact graded. And we saw that gradation in terms of those fingers from thin to thick. And here's a sort of classic version where we have an annotation here from normal to glandular to solid to mucinous, all of which are actually intermixed in this particular specimen. We went through and found the individual histotypes. Our PATH team led by Sandro and Shannon Coy went through and did that. We could then build a machine that could find that, a machine learning tool that could actually find all of those classes, allowed us to quantify them. Normal, and then you're seeing over on the right here, this is the tumor region of that specimen. You can see the scale again, a whole slide image. We've suppressed the muscle region up at the top. And we've also suppressed down at the bottom the lumen of the colon. You can see normal is normal in here, but all the rest of the tumor actually seems to be a gradation of phenotypes as represented by the Shannon entropy. And when we go in and we actually look at a molecular level, and this is still sort of a preliminary analysis, I don't think what I'm showing you here is anything other than diagnostic of these transitions. I don't think it's causal. But what we actually see is that what you tend to think of as discrete phenotypes are in fact actually intermixed on a spatial scale of around 100 to 1,000 cells. Here's an example. We have a region of normal to glandular and poorly differentiated going in the opposite direction. And you can see over here on the right that it actually correlates remarkably well with molecular gradients that we can see by imaging. And we've seen this repeatedly through all of the specimens we've looked at. What appears in the transcriptional data to be discrete phenotypes, examples of archetypes really, poorly differentiated or glandular or normal, are in fact intermixed on these spatial scales of hundreds of cells and actually appear as gradients. And the analogy we'd like to draw here are to the gradients which are already known to pattern tissues in development. So we think, I think because of the sort of way genomic analysis goes, these are discrete states. But in fact, we see these gradients on spatial scales for about a third of the markers that we've looked at. So let me finish then in my last section of the talk to tell you actually a sort of more in-depth biology story where we've taken these sort of tools and our ability to now do whole slide imaging. And we're now looking to try to understand the very earliest stages of melanoma development between the pre-melanoma, the precursor fields, which are thought largely to have epigenetic change, through the radial growth phase into what is then the much more dangerous vertical growth phase where cells become invasive down to stage 2 and 3 and eventually on to metastatic disease. And with David Liu and others, we're looking at transit melanomas and trying to understand what actually some of those events are. These specimens are almost as a diagnostic of necessity available as FFPE, and they can be very, very small. So you need a tissue-sparing method. Working with George Murphy's dermatopathology team and Christine Lian, we found specimens in which we'll actually have multiple pathological phenotypes present in a single specimen, and it gives them some possibility of creating a progression series. And then we will actually take that series and look for epigenetic and genetic drivers, for different features of the immune surveillance. And ultimately, the hope is that we can distinguish dangerous from benign lesions, and particularly in these early stages, that subset of those lesions, which even though they would appear to have been successfully resected, occur and give rise to metastatic disease much later on. The image-centric profiling has the outline that I showed you before. We start with histopathology that allows us to look over the tumor banks available to us. We then do some of this high-resolution 3D imaging. We do on the order of 20 to 60 plex CySIF to be able to enumerate cell types, microregion transcriptomics, and then 3D reconstruction. And the specimen I'm going to talk about today, we've looked at several dozen in this way, but the one I'm going to focus on has both an exophytic region here, sort of a big region sticking out, labeled EM. It has an invasive region and what's called a brisk TIL response, which means that you've got a fair amount of immune invasion. We also have some regions of regression and also a melanoma in situ, all contained within a single specimen. So that's the comparison that we're trying to make. What you learn when you compare many specimens in single cell imaging at a numeric level here, so this is single cell type calling, shown here in UMAP plots, is actually what you also see in single cell RNA-seq, which is that tumor cell types separate by patient. So there's very strong confounding by patient identity, but as you would hope in a well-conceived data set, the immune cell types cluster across all of the individual patients, telling you you don't have strong batch effects. If we simply take the intensity of the markers in here, what we see remarkably is the cell type actually already tells us an enormous amount about spatial morphology. So here's just a sort of a simple k-means clustering with k equals 5 into 5 clusters. You can see the cluster number 3, for example, corresponds to a high level of the S100B marker, which is used as a marker of melanoma. And when you look over in the primary image on the right, at here now, it's an MITF high state. You can see that once again, it's one of these graded phenotypes, so not discrete at all. In other cases, individual spatial clusters Here you're going to see a highly distributed phenotype characterized here by being cyclin D high. But when we look at it, what we see is it's actually a little region of invasion surrounding the CD31 positive microvasculature. So the molecular profile gives us a sense of what it is in terms of cell type, but these spatial features tell us what it is in terms of the underlying histology. And we're now learning how to recognize these larger scale features using both machines and humans. Ajit Nirmal, who works jointly between my lab and the Weinstock group, took the totality of this data and worked out how to actually find recurrent cellular features here. And what he was looking at things that came both from the molecular phenotypes in terms of intensities, but also from spatial gradation. And the methods he's used come from things like ecology, for example, where you're trying to look at these spatial gradients, and there's an entire field of them. And let me show you two of those methods. The first of them is known as shift lag analysis and allows us to look for recurrent features that live within the tumor region. And what you're looking at on the left now are cell type calling for different immune cell types and different features. And we can see this sort of remarkable layering exactly where we get the invasive margin of this tumor pushing down into the cell, into the normal tissue. And we see both characteristic features at the MHC level and also at cell cycle. And we can then look at the tumor extrinsic features using a method known as latent Dirichlet allocation that looks for cell type patterns. And we can see now this characteristic feature that the immune cell is lined up right along the border of this tumor with what's labeled here as RCN9. And again, we can find recurrent features and begin over time to relate those to diagnosis. When we look across the progression axis within the 70 fields of use in 13 patients, we can go all the way from normal to a precursor field, distinguished already, even though there are very, very few abnormal melanocytes, by substantial immune invasion, this extremely complicated state we get by the time we get to a melanoma in situ, in which we see both immune activation and immune suppression within the same region, and often only a couple of millimeters apart. So let me just focus on the unique properties of this invasive boundary, a recurrent feature that we found, and we're now looking at real image data here. So here you can see the tumor region shown in blue, CD11C and ERF1, showing us that there's an inflammatory environment right on the edge. This is where we find both T cells and macrophages. And you can see here for SOX10, which would be a tumor marker, the right-hand panel, you can see that just as the tumor falls off, we actually see this very, very sharp spike of ERF1, which we take as evidence of an interferon activation. And we can in fact see that interferon signal and the cytokine response that it elicits using transcript profiling data down below. So a spatial feature that's extended over many, many millimeters in this tissue, but is only 3 to 5 cell diameters wide. And we find a whole series of those, and we can actually then go into that boundary here between the T cells and the tumor cells and actually look to see if we have immune cell interaction. And the functional signal we look for is formation of immune synapse or a PD-1, PD-L1 co-localization of the receptor and the ligand between a tumor cell and a cytotoxic lymphocyte. And in the picture I show you here, we also have remarkably engagement of a Treg as well. So enumerating these features both in sequence space and then in biology space. And that leads us to this sort of beginning sense of what's happening in the progression across this first set of patients. At the precursor stage, we already have some of these epigenetic marks coming up, 5-HMC, for example. We see an initial immune response that seems to have the characteristics of a relatively normal response, seems to be active, and we actually see T cell engagement with tumor cells. By the time we get to a melanoma in situ, still in the radial rather than vertical growth phase, we'll already see the formation of a myeloid niche. And this myeloid niche is the primary PD-L1 positive expressing cell type. It's not the tumor cells at all. And we see evidence of T cell exhaustion based on marker expression. And then finally, when we get to the invasive melanoma, only a couple of millimeters in diameter, we see sort of this fully evolved phenotype where you have an internal core of the tumor, a thin, what's shown here in bright sort of yellowish brown, a thin inflamed region that goes along the margin that exactly engages as PD-L1 dendritic cells and macrophages, and they in turn then seem to be part of the mechanism of T cell exclusion. And here's sort of what that program then looks like, mapping this out. And here's the picture I showed you before, repeated with some additional markers down below. So we've seen at least 8 of these sort of programs in action in this one small region of the tumor. We can see reciprocal exchange. We think of things like interferon, which based on the transcript profiling seems to arise in the stroma and seems to be then sensed right at the interface and within the tumor. We also believe that signals like MIF1 are able to originate in the tumor cells and actually go out and influence Treg engagement out in the stroma. And then we see this very, very strong PD-L1 cytotoxic lymphocytes are largely engaged with dendritic cells and macrophages and are excluded from the interface. There's some CTL engagement here right at that interface as well that I didn't show you, but those seem to be cells that are actually expressing fairly powerful immunosuppressive programs. So these are all phenomena that have been shown before, and I think what's new here is to see how they play out in this very, very thin and highly spatially restricted region. The other thing we can see in these specimens is that right next to this melanoma in situ that's undergoing invasion and growth, we see successful programs of inflammatory regression. And here we can actually see the normal immune cell program come along and actually fully clear away macrophages. And we see some evidence of dead and dying macrophages, but in general, these are very, very immune rich and melanocyte or tumor cell deprived regions, and they're coextensive with the tumor. And so the key question to ask now is really what distinguishes these two regions in terms of molecular programs. And that's, as you can imagine, a focus of the ongoing research. So the very last thing I want to say before wrapping up and thanking the people who did this is to sort of address the question that I started, which is how do you actually make the data I showed you intelligible and shareable? It's sort of nice, I hope, to see some PowerPoint slides, but there are a lot of pictures here. I told you each of those conclusions comes from 10 to the 6 or 10 to the 7 cells. Is there any way to actually get access to that? In work with Sandro's lab, we sort of thought about the way this is done in current practice, shown here. Actually, I think I took this from Hopkins, and it typically involves a multi-headed microscope. So it's still sort of physical interaction between an attending and her students or colleagues, as shown here. But what's really accelerated in the COVID era is the development of a series of tools for actually looking at complex pictures online. And we took inspiration from this project Mirador. And we've built now a set of tools that we call Minerva, and you can get at them through this tiny URL, or you can email me and I'll send you the links. And this actually just runs directly in your browser, and you can step through this little animation of it. You can step through as many channels as you choose. You can pan around on this thing as though you were on Google Maps. And then you can see the annotation that we want you to see as though you were in a museum and being guided through from one feature to the next. And you can see both analysis, et cetera. So as we try to move towards these complex sorts of phenotypes, we're trying to work out how we can add this to journals as online animation, so you can get a sense of the spatial organization yourself. And we have a whole library of these. I think we have over 100 of these now. If you're interested, I can send you the link. So let me just wrap up by say something about where we are from a technical standpoint. A whole lot of new technologies. I showed you a couple of those. A lot of focus on instrumentation at the moment, but it's really data analysis, which is going to become the primary concern now. And the absence of central repositories is going to be a complicating factor. I told you how spatial correlation imposes a whole new constraint on our thinking about statistical power, and that it is not going to be possible to do enumeration, never mind looking at complex arrangements using regions of interest or TMAs. There's no statistical foundation for doing that. There's a lot of challenges remaining when you can combine data from different modalities. I showed you examples where imaging and single-cell RNA sequencing were congruent or at least complementary, but as a general rule, they're not the same. A protein is not the same as an RNA, so we're gonna have to learn how to learn meta features from those things. And we're particularly interested also in ultimately in some sort of merger of research and clinical implementations, and are working on some new instruments that we think would be suitable for putting into a histopath core, such as the one that Scott runs possibly later this year. High-level biology conclusions. I think we've been surprised to see how large many tumor phenotypes really are. They're very, very complex. They almost, they look like hills and valleys. They vary continuously through space. These are not discrete features. They don't correspond to single markers, but are encoded in high-dimensional space. I showed you an example of that with EMT, and then I showed you an example of a highly spatially restricted feature that was very, very long, but very skinny, and that was operating at the invasive margin of primary melanoma, and we think is part of the suppression needed for that disease to spread. So I want to close by thanking the melanoma team, a whole bunch of individuals here. I'm going to go through it quickly, the colorectal team, and the software and data analysis team. And we're part of a much larger group, and we'd be very happy to work with anyone who's interested in these sorts of topics. Thank you very much. And I'll take questions now.
Thank you very much, Peter, for the wonderful talk. I guess, please leave any questions in the Q&A, and I'll be happy to convey them to Peter. So maybe I'll kick it off myself, Peter, with a question. So I'm curious on how you guys identify these features or programs, for example, the budding tumor cells or these interactions between the T cells and the tumor cells. Is that done in an automated way where you're feeding, I don't know, healthy examples and pathological examples and asking some form of machine learning algorithm to find these features, or is it done through just people going through them and just looking at it and discovering these?
Yeah, so it's a great question. So let me say that if we get data in from transcriptional profiles, about an hour and a half later, we have information on the recurrent features in there using things like gene set enrichment or various more sophisticated Go annotation, et cetera. So in that case, as you would imagine, Hans, it's data driven. In the case of these histological features, it still takes us months. And so it's typically driven from a feature. Let's take tumor budding, for example. There's an entire literature on it. It's actually used diagnostically in some settings. And we then looked for that in our images with our eyes. Having done that, though, we could build an algorithm to find all similar examples. And you would recognize that as an example of supervised learning. And we could do that either by looking effectively, we could either train it on individual examples, or we could go through and look at sort of a kind of like tessellation number and actually calculate that. So typically the machines help us in that case, but it's a supervised or semi-supervised approach. In the case of something like PD1 or PDL1, we're obviously just influenced by what we think is important, and we look for that feature. So it has that bias. And obviously now flipping this around over the next few years into what you can do with single cell transcript profiling, where you don't have to look for a feature, you can discover a feature, is going to be an important transition, and we're not there yet.
Mm-hmm. Thank you, Peter. Now going to the Q&A. So there's a question from Gordon Freeman. Do you see tertiary lymphoid structures, and what is happening in them?
Yeah, so the example I showed of the tertiary lymphoid structure was in colorectal cancer, where you would expect to see them, and we can certainly enumerate them. In other settings, particularly in breast cancer and work with Jennifer Guerrero, there's a lot of interest in understanding those tertiary lymphoid structures. So we see them everywhere. We can enumerate them. They're a great feature to automate. They actually tend to show radial symmetry, but I don't have anything particularly insightful to say about them, but I'm happy to email Gordon once we do.
Another question, I guess, from Angad Singh. You mentioned that tumor cells from different samples tend to separate out in general for single cell data. Would you expect the same in spatial profiling?
Yes, I think that's what. So I think, so let's go to the better developed area in single cell dissociative single cell sequencing. When you do that, as everyone knows, and I think Anand does, you see that things separate, the tumor cells separate one from the other by patient type, even though we would recognize them as tumor cells. And there's a lot of work in trying to work out what those, think of it in PCA, what those sort of loadings are. How do we get all the tumor cells to lie on top of each other? We don't know what those features are in spatial analysis. We see the same phenomenon, however, that by eye, you and I would look at a tumor cell, say in 20 colorectal cancer specimens, and we'd see them all as tumor cells, but a machine will actually cluster them away from each other for reasons that whose biological underpinnings remain to be determined. And that's typical of any single cell method where you're looking at 20 or more features. And I think some of it is artifactual and some of it is real. And we're still working on that problem. Fortunately, the immune cells don't do that. We actually look for them all to co-cluster. That's actually also a criterion in dissociative single cell sequencing that they should look approximately the same, particularly if they're from adjacent normal tissue. So it's not a purely a sort of technical artifact.
Mm-hmm. And I guess I had a question about definition of cell types, Peter. So it seems like you're taking the spatial data, ignoring space, and then doing, I don't know, teasing and some KNN clustering, defining the cell types, and then mapping them back onto space and asking if something interesting is happening in terms of spatial correlations or how the cell types are distributed in space. Should there be a notion of cell type that comes fundamentally from spatial data? For example, based on interactions or proximity. So instead of using our kind of conventional ways of defining cell types, that this data will even give us new insights on how cell types should be defined.
You're absolutely right, San. So what I showed today, which is sort of the norm in the field, is basically crackpot, in the sense that we're taking a microscope, which is fantastic at morphology and not so great at intensity. And we're using it like a lousy flow cytometer, which is to say that we individually segment cells, we calculate the intensity of a feature, and then we use that just like, if this was CyTOF data. And we've been doing that because, A, that's the norm in the field. So I guarantee reviewer 1, 2, 3, and 4 will ask for it. Secondly, it makes a very nice match to single cell transcript profile. But ultimately, that is not the way this should be analyzed. And it's not the way a human would analyze it. And so what we would like to do is just go in and directly look at the underlying features and define cell type in terms exactly as you define marker expression, location, which is very informative, and morphology, including nuclear morphology. And in principle, you can do that with modern systems at the pixel level, just pixel level machine learning. The problem is we come out with an answer that's then difficult to relate to sort of kind of single cell data that comes from other omic methods. So somewhere in there is going to be a hybrid. And that is, I think, the pivot that absolutely has to happen this year. And when that happens, you'll realize, and multiple groups have done this. There's some beautiful work from Ann Carpenter showing that 5-color morphology, this is of tissue culture cells, has the same information content as RNA-seq, which is seemingly much higher dimensional. So here, I think we are ignoring the vast majority of the dimensionality of this data by not using morphology, and absolutely acknowledge that as a problem. And I can tell you everyone's working on it, but if anyone out there in the audience has a great idea how to tackle it, we will send you the data tomorrow.
So a question from Yan-Yu Chen on the chat, Peter. Any spatial analysis studies available for GE, GJ cancers? Would be nice to see the difference of squamous versus adenocarcinomas. Yes,
so the answer is we haven't looked at that very carefully yet. I think the way, so to give you sort of the trajectory of our lab's work on this and many other labs is, we developed the imaging method SciSif about 4 years ago. And then we went off and started imaging, every tumor under the sun. And of course, what happened was we got stuck in all the same problems. So we've primarily, in the work that I showed you, looked at melanoma and colorectal. We've also done a lot of breast work with Beth and Jennifer Guerrero and their colleagues. But in principle, the methods I showed you, I think by early next year, we'll be at the stage where going back to many different types of tumors. And there's absolutely no reason why gastric versus gastroesophageal cancers shouldn't be comparable. What's available in the literature is almost entirely small fields of view from MIBI or the early generation methods. And I think we would now question the ability of those to make comparisons. However, we do have available for anyone who's interested a so-called EMIT data set that has 16 cancer types, 16 normals, and a bunch of non-cancer diseases, and that has a whole series of gastric and gastroesophageal cancers in there. So sort of to give you a sense, will it work technically? The answer is yes. Have we done anything interesting? No.
A question from Dan Zhao on the chat. As EMT was noticed, were there cells found to have both myeloid markers and epithelial markers? Which markers did you use for macrophages? Any comments on the myeloid cell markers?
Dan, so in the epithelial, yes, we end up showing both, myeloid and epithelial markers overlap. They both show sort of graded phenotypes. We're still having difficulty imaging in human tissue specimens. Some of the key things like twist, things that you would like to look at are problematic. That's an evolving area. We have a lot of macrophage markers. Dan, if you send me an email, we're happy to share with you our antibody list. Everything I showed you today is in principle available publicly on our website, scisif.org, which is soon going to be harvardtissueatlas.org. But if you just email me, you're welcome to look. And if you have ideas for better markers, we're happy to use them. In the data I showed today, we use relatively limited markers, just because in the three-dimensional reconstructions, we were only doing 25-plex imaging. So we couldn't do a lot of discrimination in the myeloid cell space.
Great. Thank you, Peter. And we are at 1 p.m. So I just want to thank you again for the wonderful talk and for everyone else for joining us for Peter's talk. Thanks again.
Thank you very much, Jahan. And I'm only a few electrons away, so feel free to email with any questions.
Great. Thank you, Peter.
Bye-bye.